Hello, my name is Carson Bannister. This is my cell project, and as you can see, I did a plant cell, and we're about to enter into this through the cell wall and the cell membrane. And the cell wall is a cellulose layer that surrounds the plasma membrane of plant cells. Because it is very porous, the cell wall is permeable to molecules and ions that cannot pass through the plasma membrane by simple diffusion. But the plasma membrane is permeable to water molecules by osmosis, but not to other molecules and ions by simple diffusion either. And ions pass through the plasma membrane by active transport and facilitated diffusion, and that requires ATP for that to happen. And over here, we're going to go through some cytoplasm and over to some ribosomes. And ribosome is composed of a large and small subunit separated by a central groove, and a strand of messenger RNA fits into the groove, and the ribosome moves along the mRNA. So here we go into the endoplasmic reticulum rough, which is an endoplasmic reticulum with ribosomes on it. And now we're going to go into the endoplasmic reticulum smooth, which does not have any ribosomes attached. And then we're going to go into the nucleus. And the nucleus is a membrane-bound organelle containing chromatin, a term applied to all chromosomes collectively when they are in a tenuous thread-like stage. And during the prophase of mitosis, the chromosomes become shorter and thicker and appear as distinct doubled bodies called chromosome doublets. And here we have all this DNA, and the nucleolus right above us. And the nucleolus is a dark stained body within the nucleus where ribosomal RNA is synthesized. And here we go back into the endoplasmic reticulum. And if you did not notice, there's the endoplasmic reticulum smooth. On the other side, it started out with the smooth and then rough, and then on this side, it started out with rough and smooth. There's just an alternating pattern on both sides with the endoplasmic reticulum. And here's the chloroplast. And the chloroplast is a membrane-bound organelle and the site of photosynthesis and ATP production in autotrophic plant cells. So this creates fluid and ATP as well. And here's the amyloplast, which is a membrane-bound organelle containing concentric layers of starch. This organelle is commonly found in subterranean storage organs such as potatoes, corns, and storage roots. An example of a storage root could be a sweet potato. And here's a mitochondrion. And a mitochondrion is a membrane-bound organelle and the site of aerobic respiration and ATP production. And energy from the step-by-step oxidation of glucose is used to produce molecules of ATP. And here we have another mitochondrion. And then we're going to go through some more cytoplasm, which cytoplasm fills the entire cell. And here's another mitochondrion, and some more cytoplasm, and another chloroplast. And here we have another chloroplast. And now we're going to go get into some Golgi apparatus. The Golgi apparatus is a series of flattened membrane-bound sacs involved in the storage, modification, and secretion of proteins and lipids destined to leave the cell and for use within the cell. It is also abundant in secretory cells, such as cells of the pancreas. So this cell, eventually, parts of this cell will just leave. And like you'll see up here, there will be some Golgi vesicles. These are parts of this organelle that have already left this and now are their own little thing that will eventually become something else. And a Golgi vesicle is a membrane-bound body that forms by budding from the Golgi apparatus, which is basically just leaving or departing. And it contains proteins such as digestive enzymes and migrates through the cell membrane. And Golgi vesicles fuse with the cell membrane and discharge their contents into the exterior of the cell through a process called exocytosis. And some Golgi vesicles even become lysosomes, which are involved in intracellular digestion. Which, if it wasn't a lysosome, if it was going to become one, it would have to be in an animal cell, because lysosomes are only in animal cells. And here's another chloroplast. And now we're going to go out through some more cytoplasm. And now we're going to leave the cell, but then we're going to go right back in. Because we're going to go visit the large central vacuole, because that takes up a lot of space and it's very important actually. So here we go. We're going to go through the endoplasmic reticulum again. And this is the rough endoplasmic reticulum. And then we're just going to skip the smooth. And here's the large central vacuole. And this is a membrane-bound, fluid-filled sac that occupies much of the volume of the plant cell. For this reason, the chloroplast, nucleus, and other organelles are displaced to the periphery of the cytoplasm around the central vacuole. In addition to the water, this large vacuole stores salts, water-soluble pigments, and potentially toxic molecules in the form of crystals. So we're going to see some of these crystals up here. Like this, for example. This is a graphite crystal. This is, from above, you'll notice that it looks kind of like a needle, in a way. And the cytoplasm in here also is different than the cytoplasm outside. It's got a different color scale and all that. And then also you have a Druce crystal, which sort of looks like a diamond from above. And on the inside you can kind of tell how it would look like a diamond. And now we're going to go through some more cytoplasm. And then we're going to go back out of the large central vacuole through the nucleus. We're going to go out through the endoplasmic reticulum. And we're just going to make our way out of here. And then we are going to leave. And we're going to look at this from above, and you'll be able to see visually some of the things I was talking about earlier containing the endoplasmic reticulum, how they alternate, and also how the cytoplasm is different than the other cytoplasm outside the large central vacuole. So here we go. Now you see the endoplasmic reticulum. On this side it goes from rough to smooth, and it goes to smooth rough from the outside in. And then you see how the Druce crystal looks like a diamond in a way. I'll zoom in. And you can see above how that looks like a diamond, and you see how these are needle points. And you can see the difference in the color of the cytoplasm. And you can see how much of the large central vacuole that it actually takes up of the cell. And you can also see the actual cell, that it looks like a rectangular shape. The edges are just a teeny bit curved, but it's not circular like an animal cell. And then you also, if you zoom in, you can see the difference in the color scale of the cytoplasm, the cell wall and the cell membrane. You can see how that's different. You can see the tops of these cells. That's the amyloplast. There's the mitochondrion. There's the chloroplast. The nucleus is the big sort of pyramid shaped in this model. And here is the endoplasmic reticulum with the ribosomes. That's the ribosomes, and then the smooth without any. Then you see the loose ribosomes over there. Then over here you see the Golgi apparatus with the Golgi vesicles already loose and broken away. You can see where they've broken away from. And then you have more chloroplast and mitochondrion. And that is a plant cell for you. I hope you guys enjoyed watching this video. I definitely enjoyed making this project. It's very fun for me. I hope to get another project like this, and thank you for watching.